Howdy, my future doctors. So today we are going to discuss about major and minor cross matching in blood transfusion. So this is a topic which got me confused very much. Okay, when I tried to find an answer for this, I couldn't find it from one place. It was scattered all around. Okay, so that's why I decided to do a video on this. Now, what is major and minor cross matching and how it is done, or what is the basis of this procedure, why it is done, and why exactly it is called major and minor cross matching, respectively. So, these are the things which we are going to see in today's video. So, let's get straight into it. First, let us consider the major cross matching. In major cross matching, what happens is we'll use donor's cell, the RBCs, and recipient's plasma. Okay, donor's cell and recipient's plasma. Remember, it is major DC. DC is a major comic book. Now, let us understand cells of donor means the red blood cells. When you consider the red blood cells, so it contains agglutinogens. If you ever confused with the terms agglutinin and agglutinogen, antigen and antibody, then just remember this, okay? Agglutinogens are antigens because both are ending with gens. Okay, is that clear? So what the plasma of the recipient contains, it does contain antibodies or agglutinins. Okay, so good. Now what happens is these agglutinogens or antigens are highly antigenic. So when it is introduced to the plasma of the recipient, this highly reactive agglutinogens will react with the agglutinins in the plasma. This results in the clumping of blood. In case incompatible, it results in clumping. If it is compatible, no clumping. Now let us discuss about the minor cross matching, and let's come back and explain why it is called major and minor respectively. Okay, good to go. Now minor cross matching, what we do is we will take donor's plasma and recipient's cells. So major is DC. Now minor we can make on our own, the donor's plasma and the recipient's cell. So donor's plasma contains agglutinins and the recipient cells contains agglutinogens. Now what happens is these agglutinins from the donor's plasma are not as reactive as the agglutinogens. So when these plasma is introduced into the recipient's body or recipient's bloodstream, what happens is it actually gets diluted in the higher volume of recipient's plasma. So it wouldn't be causing much of clumping problem. It will cause clumping, but not to a degree of clumping caused by the donor's cell. Okay. Incompatible will cause clumping. Let us put an arrow here which shows a low degree of clumping. And compatible means, of course, no clumping. Okay, so this is what happens in major and minor cross matching. Let's go through this once again. In major cross matching, we use donor cell and recipient's plasma. Donor cells or the RBCs contain agglutinogens and the recipient's plasma contains agglutinins. Agglutinogens are really antigenic. Okay, so this will cause a major clumping reaction. Okay, now for why this reaction is called major cross matching. In this reaction, even a small amount of donor's RBC is present or transfused to the recipient's body, it will cause clumping, a lot of clumping. That's why it is called major cross matching, because it plays a major role in determining whether this blood group is compatible or incompatible. Now, for minor cross matching, we use donor's plasma and recipient's cell. So what happens is when the donor's plasma reaches the recipient's bloodstream, already the antibodies in the plasma are cold antibodies. They are not as reactive. Okay. and also it is being introduced into a larger volume of plasma in the recipient's blood, so it will automatically get diluted in that blood, and which is not responsible for a major clumping reaction. So it is not as important or not as indicative for this compatibility and incompatibility of blood groups. Let's wrap it up once again. Major cross matching, donor cell and recipient's plasma. Donor cells contain agglutinogens. Recipient's plasma contains agglutinins, antigens and antibodies respectively. So these antigens or the agglutinogens are highly antigenic, and even a small quantity of this will cause clumping. And if this happens during transfusion, it can cause major problem for the recipient. So this process of cross matching is very important in determining the compatibility between blood groups. That's why it is called major cross matching. Now coming to minor cross matching, the donor's plasma is combined with the recipient cell. So the donor's plasma, it has agglutinins, and the recipient's cell contain agglutinogens. So when this plasma is introduced into the recipient's system, this plasma will get diluted. Along with the fact that plasma has cold antibodies, the reaction or the clumping will be very low compared to this major cross matching. So it is not as important in determining the compatibility between two blood groups. That's why it is called a minor cross matching. So I believe you guys understood this concept. Read it again and you will get a better idea. Okay, hopefully it's not confusing anymore. Thank you guys for sticking around. Let's see you in the next video. Bye.